Welcome grade 11s. I want to welcome you to the Junior Tikkie Summer School. So there will be a series of videos which will be live streamed in the next few weeks on our Junior Tikkie.online platform. Please use the given material as revision and also look at all the grade 11 content on our platform. So let's get started. I am Ms. Kutsia, and today we will look at grade 11 life science papers from November 2021. So let's move on to question number 3. So remember, question number 3 is part of section B, and this will be a total of 50 marks out of the 150. So question 3 is regarding a graph that will show the relationship between the light intensity and the release and the uptake of the carbon dioxide. So this is a very important section because here we have two dependent variables that's indicated on one graph. So that will also give you one of your variables as well as the light intensity. So on your y-axis, you will find your dependent variables. So in this case, you have 2, which is the take-up of carbon dioxide and also the release of carbon dioxide. Then you also have the light intensity, which is your independent variable and that you will find on your x-axis. So let's have a look at the trend of the graph. So as we look at our axis over here, you will see that there's an increase in the light intensity. So if we have a look at the graph, I'll quickly use an indicator. You will notice that as my light intensity increases, there will be a sharp increase and a quick increase in the uptake of carbon dioxide. So that gives me an indication that the rate of photosynthesis will be quite quick. And then about at a 30, it will reach a constant uptake. So that means that the rate of photosynthesis also stayed constant here. There will be no increase. So we can actually use this to our advantage because we have an excessive increase in carbon dioxide level. So that will actually in light intensity as well as our ozone layer is depleting. So if there is an increase in my light intensity, there will be an increase in the uptake of carbon dioxide. And then even though I increase my light intensity even further, that means that the uptake of carbon dioxide will remain the same. Then if we look at the increase of light intensity versus the carbon dioxide release, so how fast carbon dioxide is released. So this now changes from carbon dioxide uptake to carbon dioxide release. So here you will see that my light intensity increases. There's a quick and sharp increase in the release of carbon dioxide, and then it stops. Then there will be no more release of carbon dioxide release. So this will give you an indication most likely that the light intensity could be too high, can denature enzymes that's responsible for cellular respiration, which then produces carbon dioxide gas in the process. So let's quickly read through question 3.1. So I've discussed the graph over here. So let's quickly have a look at the specific questions. So question 3.1.1 asks, at what range of light intensity is carbon dioxide released? So you will go to your graph, you will look at the rate at which carbon dioxide is released. Oh, this one is taken up. So we'll have a look here at the bottom and then compare it with the light intensity on the x-axis. Then question 3.1.2, name the process taking place in the leaves which uses carbon dioxide. Then question 3.1.3, why is most carbon dioxide released when the light intensity is at 0? When the light intensity is 0. Then 3.1.4, how much carbon dioxide is taken up by the leaf at the light intensity of 20. So you'll go to your graph, you will look at 20, and then you will compare it with the carbon dioxide that is released, that's taken up. So that you will look at the top part of your graph. Then question 3.1.5, when the light intensity is approximately 2 units, there is no net change in the concentration of carbon dioxide surrounding the plant. Give an explanation for this. Then 3.1.6 at the light intensity above 30. So above 30, that section over there, the amount of carbon dioxide taken up remains the same. Suggest an explanation for your observation. So let's have a look at our memorandum of question 3.1. So question 3.1.1 asked, at what range of light intensity is carbon dioxide released? So if we have a look over here, if I just get my laser over here, you will see this area over here. So they say anything from 0 to 2 arbitrary units. Then they ask you, name the process taking place in the leaf which uses carbon dioxide. So where it is taken up. So that's this section. So that will be photosynthesis. Then, why is most carbon dioxide released when the light intensity is 0 units? So that will be over here, and we will focus on the section over here. So where the light intensity is the least. So the reason for that is because there's no photosynthesis taking place, and only cellular respiration is taking place. Then question 3.1.4, how much carbon dioxide is taken up by the leaf at a light intensity of 20 units. So if you find 20, you read upwards and you give me a reading there of 6 milligrams or even 6 milligrams per 50 centimeters cubed. So please remember that unit, that's very important. Then question 3.1.5, when the light intensity is approximately 2 units, there is no net change in the concentration of the carbon dioxide surrounding the plant. Give an explanation for this. So they say if we look at the light intensity when it is about 2, then they say there is no net change. So the reason for this is that the rate of photosynthesis is equal to the rate of cellular respiration. So that is why it will seem at this point that there is no net charge of the surrounding carbon dioxide levels. So at this point here at 2, we will see of a light intensity of 2 that the rate of photosynthesis and respiration is equal. Or you can also say the amount of carbon dioxide that is produced is then also used during cellular respiration. That the carbon dioxide that is produced during cellular respiration is equal to the amount that is used for photosynthesis. Then question 3.1.6, at the light intensity above 30 units, so above 30 units, the amount of carbon dioxide taken up remains the same. So here you will see that we reached a constant uptake. So suggest the explanation for this observation. So that means the rate of photosynthesis remains constant because it's now reached an optimum concentration of carbon dioxide for photosynthesis that has to be taken up. And also because chlorophyll molecules were saturated and enzymes are limiting factors. So let's read through question 3.2 and look at the memorandum. So firstly, they have given us the section of the human lung, specifically the whole entire breathing system. So firstly, they ask us in question 3.2.1 to identify parts D and E. So if we have a look over here, we will see part D over here specifically refers to my intercostal muscles, and then E you will find on this side, which is the diaphragm. Then they ask us to name the epithelial tissue that lines the inside of part C. So part C over here is my trachea, where part B will form part of the larynx. And then the next section states the function of label B, but you will see that I've added a correction over there, so it asks then for C. Then 3.2.4 describe the process of inhalation. So you just need to describe the changes that takes place when I am inhaling. So let's have a look. Part D is your intercostal muscles. Part E is the diaphragm. Then the epithelial cells that lines C, so that will be your columnar or your ciliated epithelial cells specifically. And then they ask state the function of part C. So that is, it is made out of C-shaped cartilage rings to keep the trachea open at all times. And then you had to describe the process of inhalation. So I'll add a little diagram for you. So you will see over here that when I inhale, the diaphragm will contract. If the diaphragm contracts, it will flatten, and that gives you 1 mark. So that the length of the thoracic cavity increases, so that it's larger, it pulls up, it opens up. Your external intercostal muscles will contract to lift the ribcage upwards. So this causes the thoracic cavity to enlarge, so the volume becomes bigger. So the total volume then increases, and the pressure on the lungs then decreases. So since the atmospheric pressure is then greater, the air will actually rush into the lungs. Then the air rich in oxygen is then drawn into the lungs, through the air passages like the trachea, the bronchus and the bronchioles, and then into the lungs. So let's look at the last section of 3.2's memorandum. So 3.2.5 said that we had the coronavirus that responsible for COVID-19. It affects the lower respiratory passages. So fluid will then accumulate in our pulmonary tubes, and then it will cause accumulation of fluid in label A as well. So if we go back to our little label over here, you will see that label A will refer to the alveoli. So then furthermore, the tissue that lines part A becomes scarred, and then it thickens as well. So let's quickly read the question. So they say, name the part that's labeled A. So again, label A is my alveoli or my alveolus, and then explain why the infection of the lower respiratory system of pathways can lead to possible death. So they started off saying that the coronavirus is an infectious disease, so there was no mark allocation for that. So this causes the squamous epithelial cells to become scarred and thickened. So this is the tissue that lines the alveolus. Then this prevents gas exchange from taking place, so between the alveolus and the surrounding blood capillaries. Then accumulation of the fluid in these pulmonary tubes, like the bronchioles, will then prevent air from flowing into the alveoli. Then there will be less oxygen available for cellular respiration, and this will then result in organ failure due to a lack of energy. So question 3.3 is our second last question of the question paper. So let's quickly read through the questions, and then I'll go through a summary that's based on what was asked in this section. So they said that the diagram below represents a certain section of the alimentary canal. So that is just a certain part of the digestive system. Then you will see that they've given you a specific diagram. Let me just show you. So here is your diagram with a few specific labels. So if we have a look at our diagram, we will see that the label includes, or the diagram includes, the liver. This little structure over here is the gallbladder. This section over here is a tube that connects the gallbladder to your small intestine. Over here we have the pancreas. This is also your small intestine, which label A represents. And then this section is the beginning of your large intestine or your colon. So let's quickly go through the questions. Question 3.3.1 says identify part A and part C. So if we have a look here, here is part A, there's the structure, and then this section over here is part C. Then they ask in question 3.3.2 to state 3 functions of the part that's labeled B. So this is the liver. There's quite a few functions for you to list over there, and it's a total of 3 marks, so just 3 functions. Question 3.3.3, explain how fat digestion would be affected if the duct that's labeled D were blocked. So over here you will see there's D, so that's if I have a blockage over there in label D. So that means it's going to affect the bile that's stored in the gallbladder, because remember the bile is produced in my liver, it's stored in the gallbladder. Then bile will move into the small intestine for the digestion of fat. But now the duct is blocked. So that means no bile can then enter into the small intestine. Then question 3.3.4, they ask you to explain 2 ways in which the part labeled E, okay, is structurally suited for the efficient absorption of digestive nutrients. So that's 4. So remember, to explain, you have to give the structure and then explain why it is efficient for absorption. So let's quickly move over to my summary of the human digestive system. So over here you will see that there's a few labels, so I'll just wait for the little picture. So food will be consumed through the mouth, will move down the esophagus. So remember, the esophagus is a muscular tube which contracts and relaxes to push the food down the esophagus towards the stomach over here. So that's known as peristalsis. We have the stomach. This little orange structure over here is the gallbladder. Then we have the liver. So remember, the liver has quite a few functions. It stores glucose in the form of glycogen. It is responsible for detoxification, so it removes or breaks down toxins in the body. It is responsible for deamination of amino acids, so breaking them down to form glucose and urea. And then it also stores things like vitamins and iron. Then we over here have the large intestine, which is mainly responsible for the reabsorption of water. And then we have the small intestine, which is quite coiled over here. It's folded into the digestive or in the abdominal cavity. So this is where where absorption takes place of most of the digestive nutrients. So let's have a look at the memo. So question 3.3.1 just asked you to identify label A, which we will see is the duodenum. So this specifically is a section of my small intestine. Then C is my, I must C over there. So C is pointing towards my gallbladder. So remember, the gallbladder stores bile. Then question 3.3.2 asks to state 3 functions of the part that's labeled B, and remember that is the liver. So here it secretes and produces bile. Then it converts excess glucose into glycogen. So that means it is glucose that's not used for cellular respiration, or it stores then glucose in the form of glycogen. So it stores minerals like iron. It stores vitamins like vitamin A, D, and B12. It's responsible for the deamination of excess amino acids. So remember, when this gets broken down, it forms glucose and urea. And then it's also able to detoxify the body and get rid of harmful substances like alcohol or any medication and drug that's taken in by the body. Question 3.3 says, how will the digestion of fat get influenced if that section, if that tube is blocked? So firstly, bile will not be able to reach the duodenum or the small intestine. So then there will be no emulsification of fat. So fat won't be broken down into smaller sections. So that means the enzyme lipase will not be able to digest the fat into fatty acids and glycerol. And remember, this is the form in which fat is absorbed into the small intestine, or specifically into the lacteal. Question 3.3.4 explain 2 ways in which part E, so part E is this section over here, is structurally adapted for its function, for absorption. So number 1, you mention the structure, which is very long. Why is it very long? Because this ensures that there's a maximum period for absorption. Then it contains millions and millions of villi. So this is to increase the surface area for absorption. Then the walls of the villi is made up of a single row of columnar epithelial cells. So this actually facilitates the fusion. Then it contains a lacteal and blood capillaries. So this is also to facilitate and transport absorption of digested nutrients. Then the columnar epithelial cells also contains villi, which is microvilli. We also mention it over on this section, but this is just the villi on its own, the villus on its own. So these cells contain its own microvilli to increase the surface area for absorption. Then lastly, the columnar epithelial cells also has a high concentration of mitochondria. So this provides energy for active absorption of nutrients. Question 3.4 is now our last question of section B and the last question of our question paper. So let's quickly read through the section and see what is asked from us before I go through the summary. So it says that in the given diagram below, you will see that they have given you an organelle in a plant cell. So the organelle is a structure that's found in a cell, and because it's in a plant cell, you can automatically, because we only have done the mitochondria and the chloroplast, it can only be one of those two. So in this section, let me just get my laser. You will see, if you look at the diagram, this will identify or give you an indication that this is a chloroplast. So most of the times when it's regarding the plant, they will ask you the chloroplast, and usually when they ask you if it is a small little insect or seeds, then the organelle tends to be the mitochondria, but you have to identify and look at the structure. So you will see the granum over here, and then also the interlamella over here that you will identify to give you an indication that this is a chloroplast. So in this section, they say that question 3.4.1 says that you need to identify the organelle. Then question 3.4.2 say name the part that's labeled B. So label B over here, you will see it's into this open area over here. Then question 3.4.3 says that name the two stages of an important biological process which takes place in this organelle. Then question 3.4.4 says describe the process that takes place in the part labeled A. So you have to identify the process over here, and then you need to describe it for 5 marks. And this takes place during the day. So that's also an indication or a clue which phase takes place there. Then the next question, 3.4.5, explain whether the environmental temperature could either slow down or stop the process that takes place in the organelle represented over here. So they just want to know what is the effect of temperature on this process that takes place. So let's quickly have a look at our summary. So on my right hand side, you will have the structure of the chloroplast. So remember, your chloroplast is where the process of photosynthesis takes place. So you will see we have 2 dark green lines over here. So we actually have a double membrane. So we have an outer and an inner membrane, and then the space in between is the intermembrane space. If we move to the inside, so this was one of our labels, so the open space over here, so this is the stroma, so this is an aqueous solution, very similar to my cytoplasm that I find in the cell. Then over here, you guys will see that I have these little three structures. It sort of looks like Smarties to me. So this is your granum. So then your granum is lined with a small little structure which is known as the thylakoid. And this is where I will find my chlorophyll, the site of photosynthesis. Then over here, I have my interlamella, or my intergranum lamella over here, and then this inside over here is my lumen. So let's have a look at the different phases. So there's two phases that takes place. So over here on this side, you'll see we have the light phase. So that's why it's lighter in color, or also your light dependent phase. On this side, you will see it's a little bit shaded, so it's a bit darker in comparison to the left. So this is your dark phase, and this is then also your light independent phase. So this phase takes place independently if there is light, if there is no light. This section can only take place if there's light possible. So just in short, radiant energy moves into the chloroplast. It moves to the chlorophyll where the light is then trapped. So the light is then used as energy to split up the water molecule. So when water molecules are splitted up, the first product that's produced is oxygen. It leaves the chloroplast, it leaves the plant through the stomata into the atmosphere. The hydrogen then gets moved to the next section, to my dark phase, where it enters the cycle. So when it enters the cycle, the hydrogen attaches to a carrier molecule, which is NADP. So when it attaches, it is known as NADPH. So then the hydrogen reacts with the carbon dioxide that enters the cycle, and then at the end, it forms a sugar specifically known as glucose. That glucose can then be used for cellular respiration, or it can also be stored in the form of starch. So let's have a look at the memorandum. So it first says identify the organelle. So the organelle is chloroplast. Then they ask you to name label B. That is going to be your stroma. Then name the two stages of an important biological process which takes place. So they didn't mention what the process is, but the process is photosynthesis. So we have the light and the dark phase, or you can also say the light dependent and the light independent phase. Then question 3.4.4. Describe the process that takes place in label A. So here you have to identify if it's the light or the dark phase. But they say that takes place during the day. So that is your clue. So over here I have the light phase that takes place. So the light phase takes place in the grana of the chloroplast. So this contains chlorophyll. The reason for that is that it absorbs the light energy. It traps the light energy. Then the light energy is used to form ATP. Then the light energy is also used to split the water molecule, and that process is known as photolysis. So remember, the word photo refers to light and lysis means to break down. So then this forms a hydrogen, an energy-rich hydrogen, and also the oxygen molecule, which then enters the atmosphere. Question 3.4.5, explain whether the environmental temperature will affect the process that takes place in this organelle. So high temperatures will then affect the protein molecules, the enzymes, because they will be denatured. So that means the enzymes cannot perform their function. They are functionless. So this causes the rate of this metabolic process, which is photosynthesis, to stop. So at first it will only slow down and then eventually stop completely. But very low temperatures then again will just slow down the enzymes. They become inactive. So that means the rate of photosynthesis will take place at a very, very slow rate. Thank you, Junior Techies. Please make sure that you watch our next videos.